This is the 5th video presentation for the CASE Animal and Plant Biotechnology course. As you view this video, be sure to make notes of important information. Feel free to pause, repeat, and resume the video whenever necessary. Plant Cloning, Lesson 4.2, Performance Enhanced Plants. You researched and compared methods of inserting genes into plants during Activity 4.2.1, Getting Genes into Plants. Once plant cells have the desired genes, new plants must be created to express the genes across a population. Tissue culture, or micropropagation, refers to the process by which individual cells are coaxed to develop into a new plant. In vitro means in the test tube. Micropropagation uses a very small amount of plant tissue that may not contain enough nutrients or hormones to support cell multiplication and formation. Agar provides water and nutrients to encourage cell multiplication. Once an unorganized group of cells is formed, an agar containing nutrients and auxins is used to force the tissue to develop into specialized structures, including roots and meristematic tissue. The unorganized group of cells develops into a mass called a callus. From there, cells will multiply and differentiate into plant tissues and systems. Any part of the plant can be used if the tissue is living. A higher success rate is experienced if young, aggressive growth tissue, such as meristems or new leaves, are used. Culturing is done from parent plant that has desirable characteristics. After taking a cutting from the parent plant, the cutting is prepared for regeneration. Clusters of embryos will have to be divided just as we did with the chrysanthemums or grasses in the Principles of Agricultural Science plant course. In a sterile environment, the embryos will be transferred to agar containing auxin, which is a rooting hormone. Hormones cause the embryos to develop structures needed for the plant to survive. Roots, shoots, and buds emerge to form young plantlets. Still susceptible to disease, young plantlets are grown in controlled environments until they can survive in normal growing conditions. Certain requirements must be met for optimal growth. These essential needs of the plant cells in tissue culture is no different than normal growing plants. The environment is altered to meet the requirements of young, tender cells. Advantages to tissue culture make it a great tool for mass propagation of desirable plants. In most species, multiplication of plants using the micropropagation is faster than sexual propagation. This process is especially useful when only a small amount of plant material is available for a desirable plant. Several offspring can be generated from just a few cells. Special equipment and skills are needed to perform tissue culture with great success. The plants are very vulnerable to injury and death in the early stages of development. As a review, mark or highlight 3 key points in your notes that are important to remember from this presentation. List 2 key ideas or concepts that relate to previous knowledge. List any questions you have about this topic. Discuss these questions with your instructor. Keep any notes you have from this presentation organized and available for use throughout the course. Through Activity 4.2.2, Culturing Ferns, you will propagate ferns using tissue culture and sterile technique in order to understand that the totipotency of plants allows minute proportions of plants to be cultured into a complete plant. Your teacher will provide more instructions. For more information about sterile technique, view the video presentation titled Sterile Technique for Plant Cloning.